we have completed about the tyrosine and tryptophan and then phenylalanine. And first metabolism that I taught you about the tyrosine and phenylalanine. And remember phenylalanine. Remember phenylalanine and tyrosine. Thank you. Phenylalanine and tyrosine. We have learned that phenylalanine will get converted to tyrosine by addition of OH group. By addition of OH group. OH group will be added into that one. And addition of OH group will be by phenylalanine hydroxylase enzyme. We have learned that one. And then after formation of the tyrosine is going to form melanin, and then tyrosine is going to form T3D4, and tyrosine is the one which is going to form the catecholamines. Is it okay? And once that is done, we have learned about tryptophan. Tryptophan is helping us in the formation of serotonin, melatonin, and one more thing, that is vitamin B3. Okay. And we have learned about two diseases yesterday. One will be the phenylketonuria, another one will be the alkaptonuria. Those two diseases we have learned yesterday, guys. So now today let us continue with our discussion right now, guys. The first topic that we are going to learn today right now will be the metabolism of aliphatic amino acids. Metabolism of aliphatic amino acids. Okay. And if you remember yesterday's class, metabolism of aliphatic amino acids. Yes, aliphatic amino acids, they are like 5 of them: glycine, alanine, and then valine, and then leucine and isoleucine. So let us learn about all the 5 aliphatic amino acids right now. What is this, guys? Sir, since there is one phosphate group in glucose 1-phosphate and glucose 6-phosphate, it is the first on one side and 6th on the opposite side. So how can we differentiate first and 6th position? Hello, iPad 2. Whatever diagram of glucose that we have drawn in our class yesterday, it was like 6 carbons like this: 1, 2, 3, 4, 5, 6. This is not the exact diagram of the glucose. I have drawn only the carbon skeleton. If you draw the exact glucose structure, you'll be easily able to identify and differentiate the first carbon and the 6th carbon. Are you getting a point here? तो हमने जो ड्रॉ किया है उसमें आप परेशान मत हो कि सर हम कैसे डिफरेंशिएट करेंगे. सो दिस विल बी द एक्चुअल डायग्राम ऑफ द ग्लूकोज मॉलिक्यूल. परफेक्ट, नाइस, वेरी गुड. ओके, तो हम डिफरेंशिएट कर सकते हैं. एंड वन मोर पर्सन इज आस्किंग अ डाउट हियर: tetrahydrobiopterin to dihydrobiopterin. Yeah, we have seen phenylalanine to tyrosine. I told you phenylalanine to tyrosine. Guys, other people, let me just take up these doubts, clarify, and then आगे बढ़ेंगे हम लोग. I told you phenylalanine to tyrosine, addition of OH group, name of the enzyme phenylalanine hydroxylase. Okay, and who is adding the OH group? OH group is added by tetrahydrobiopterin. There are 4 hydrogens, and after adding the OH group, it is getting converted to dihydrobiopterin. Okay, I hope that is clear. So phenylalanine is getting converted to tyrosine by addition of OH group, and OH group is added by tetrahydrobiopterin, and after adding the OH group, it is becoming dihydrobiopterin. Okay, chalo. If these doubts are clarified, welcome to our today's topic. Heading लिखिए sir, metabolism of aliphatic amino acids. Aliphatic amino acids will be your glycine, and then we are having alanine, and then valine, leucine, and isoleucine. First, let's get started with the glycine here right now, guys. चलिए sir, glycine start करते हैं. ठीक है? Now first let's get started with our today's topic right now, that is glycine. Okay, now my dear friends, glycine the first point to remember about glycine is, out of all 20 amino acids, it is the simplest amino acid. Out of all the 20 amino acids, simplest amino acid is glycine. Number 2, second thing is that, remember, it is the most abundant amino acid. It is the most abundant amino acid in collagen. Recently asked question. So in the collagen protein, the most abundant amino acid will be your glycine. And then the third point that you have to remember is the flexibility of the protein. The flexibility, or we can say the bends in protein. The flexibility or the bends in protein are due to which amino acids? Are glycine amino acid. So glycine is actually the simplest amino acid. Glycine is simplest amino acid out of 20, and it is the most abundant amino acid in collagen protein. And in the proteins, whatever flexibility you are able to see, or whatever bends you are able to see in the proteins, those bends or the flexibility in protein is due to which amino acid? It's due to glycine amino acid, guys. Okay. Now once you are done with this one, let us see what are the functions of glycine. Glycine के ना functions क्या होते हैं सर? What are the functions of glycine, guys? ठीक है? Divya is asking something. I'll get back to it, Divya. Okay now. Now, glycine. Let's see what are the functions of glycine right now, sir. Glycine के functions देखते हैं. Function number 1, glycine helps us in the formation of heme synthesis. Glycine helps in heme synthesis, guys. Okay. When you study about the heme synthesis, I'm not telling hemoglobin. I'm not at all telling you about the hemoglobin. I'm telling you about heme. Okay. When you study about heme synthesis, the very first reaction in the heme synthesis is succinyl-CoA. Succinyl-CoA combines along with the glycine, and these two will combine together to form your delta-ALA, and delta-ALA will undergo n number of reactions there, and after that it is going to form heme. Okay, so I'll teach you about the heme synthesis topic later on, guys, but right now the heme synthesis, we need which amino acid? Glycine. Glycine is the amino acid required for heme synthesis. Done. Next, number 2, glycine is also required for purine ring synthesis. Not the pyrimidine ring, purine ring synthesis. We require which amino acids? Are glycine. So don't worry about this. Tomorrow we are going to study about the DNA and RNA wala part. In that I'll teach you about the purines and pyrimidines. And the purines will be your adenine and guanine. And the formation of the purine ring, that is adenine and guanine ring, you require what's a glycine. और मैं आपको ना उस topic में पढ़ाऊँगा. Don't worry about that. Number 3, glycine is also required for creatine synthesis. Okay, creatine synthesis. Glycine is also required for synthesis of creatine. Okay. And number 4, glycine is also required for the synthesis of glutathione. So yeah, in short we can say that it is required for synthesis of many things. So glycine is required for heme synthesis, for purine ring synthesis, for creatine synthesis, as well as glycine is required for glutathione synthesis. And now they want you to remember in detail about glutathione. Glutathione is actually a tripeptide. Glutathione is tripeptide. What is the meaning of tripeptide? It is made up of 3 amino acids. And sir, how to remember the 3 amino acids? I'll tell you the trick here. First of all, gluta. That is the reason why which amino acid here, sir? That is glutamate. And the second word here will be thio. I hope if you have learned a little bit of chemistry in your life, thio is the word used whenever you have got the sulfur containing amino acids. Okay, so wherever you have sulfur, the word used will be thio. So therefore sulfur containing amino acid, that is cysteine. And finally one there, and the first one amino acid, pehla wala, and that is glycine. Is it okay? So I remember it something like this. Are you getting me? So glutathione, that is glutamate, and thio means sulfur containing cysteine and glycine. Remember in this exam, MC exam, they have already asked about this one, guys. You already पूछा गया था. Okay, glutathione is made up of how many proteins, how many amino acids, guys? 3 amino acids, that is glutamate, cysteine and glycine. Glutamate, cysteine and glycine. Is it okay? So glycine is required for heme synthesis, glycine is required for purine ring synthesis, as well as creatinine synthesis, as well as glutathione synthesis. Okay, and after that write down function number 5. Glycine is also acting like a neurotransmitter. I think you have learned physiology, and in physiology when you learn CNS physiology, in CNS physiology now wherever there will be a synapse, there will be one neuron and then there will be another neuron, and between the 2 neurons there will be a gap here, and that is known as synapse. In synapse there will be release of a chemical substance, and that chemical substance is known as neurotransmitter. And there are variety of neurotransmitter. We have got norepinephrine, you have got like so many of them, acetylcholine, GABA, glycine. So even glycine also acts as a neurotransmitter. Glycine also acts as like a neurotransmitter. And function number 6, glycine also helps in conjugation of bile acids. bile acids. Bile acids will be produced by liver. Liver will be forming the bile acids, and after forming the bile acids, those bile acids has to be conjugated, and that conjugation of bile acids will be done by glycine. So glycine also helps in conjugation of bile acids. So these are all the functions of glycine that you have to remember for your exams, sir. More than enough. Done. I repeat again, glycine is required for heme synthesis, glycine is required for purine synthesis, glycine is also required for creatinine synthesis as well as glutathione, guys. And glutathione is a tripeptide. You have to remember in detail, made up of 3 amino acid, glutamate, cysteine and glycine. And then glycine is also acting like a neurotransmitter and is also required for conjugation of bile acids. That is all about the glycine, guys. Now let us go to the second amino acid. The second one will be your alanine. And my dear friends, nothing to learn about alanine. Why? Conjugation is like combining. Okay, we are going to conjugate glycine along with the bile acids. Arjun, doubt what is conjugation? Conjugation means joining. So glycine amino acids will be joined by the bile acids. Okay. Now welcome to the alanine, guys. Alanine we don't have to learn. Why? Because we have already learned glucose alanine cycle. Okay, ji. So we don't have to learn it again. Already I have taught you that in the previous topic itself. So what is the function of alanine? You remember glucose alanine cycle? Muscle to liver, then again from the liver back to muscle and all those. So that entire cycle is nothing but glucose alanine cycle. That is the function of alanine. And now welcome to the last three. Okay, we are done with glycine, we are done with alanine, and now valine, leucine and isoleucine. Remember, these three together, they are known as BCAs, the branched chain amino acid. Okay, branch chain amino acids कहते हैं, ठीक है? So please write down the subheading there as the branched chain amino acids. And which will be the branch chain amino acid? One will be your valine, another one will be the leucine, another one will be the isoleucine. And these three branch chain amino acid, valine, leucine and isoleucine, will undergo certain metabolism and they are going to form some product. So the good news here is that you don't have to learn the entire pathway. You got to learn only and only about one enzyme here right now. What is the name of the enzyme? It's branch chain. Okay, why? Because these are branch chain amino acids, branch chain. And if you remember, yesterday I told you one thing, the amino acids after coming inside our body, amine part will be removed off, and then what is remaining? We are remaining with the keto acid. So therefore the same thing is happening here also. Branch chain, what is left out? Keto acid. And they are undergoing oxidation. Oxidation means removal of hydrogen, and that is why dehydrogenase. So what is the complete name of the enzyme, sir? What is the complete name of the enzyme? Branch chain keto acid dehydrogenase. Branch chain keto acid dehydrogenase. Okay. So I repeat again, these three are the branch chain amino acid, valine, leucine, isoleucine. These three are branch chain amino acid, and for the metabolism of these three we require an enzyme, and the name of the enzyme is BCKD. Okay, don't take me wrongly, it is branch chain keto acid dehydrogenase. Branch chain keto acid dehydrogenase. And now this enzyme, you have to remember like 2 points here right now, guys. Point number 1, this enzyme requires coenzymes. इसको ना coenzyme चाहिए. And how many coenzymes are required for this one? Again the same thing, 5 coenzyme. Again the same 5 coenzyme. Number 1, lipoic acid. Number 2, thiamine pyrophosphate. Number 3, coenzyme A. Number 4, NAD plus. And number 5, FAD. Are you getting my point here right now? Hello my dear friends. Enzymes which will be requiring the same 5 coenzymes. Number 1, we studied pyruvate dehydrogenase complex, and number 2, we have learned about the alpha ketoglutarate dehydrogenase complex. One is PDH complex, another one is the alpha ketoglutarate dehydrogenase complex. Come on my dear friends, just confirm me. Hello, are you able to recall these 5 coenzymes? These 5 coenzymes we have learned till now. It is required for how many enzymes? It is required for, yes, 3 enzymes till now. Keep on making the list. Wherever you're writing your notes, at the last page of the notes, just make a note of that one. Okay, now 5 coenzymes required for how many enzymes till now we have learned. आपका responsibility है, make the list. And now, my dear friends, yes, welcome here right now. This enzyme is utmost important. Recently, in the last 3-4 exams, continuously they have asked the question. Why it is important? You know why? Because in some people there will be deficiency of this enzyme, and if this enzyme is deficient, then what is going to happen? It will lead to a disease. Clinical correlation. What is the disease? What is the disease? It will lead to maple syrup urine disease. Maple syrup urine disease. ठीक है ना? Maple syrup urine disease. और मैंने आपको क्या बताया था? Any time in biochemistry when you are learning any disease, first of all write down the enzyme deficient. Now what is the enzyme deficient in maple syrup urine disease? BCKD, branch chain keto acid dehydrogenase. The enzyme deficient in the maple syrup urine disease is branch chain keto acid dehydrogenase. Branch chain keto acid dehydrogenase. अच्छा, my dear friends, what is the clinical feature? इसको ऐसा क्यों कहा जाता है? What is the meaning of maple syrup urine disease? The urine smells like maple syrup, odor. And how is the odor of the maple syrup, guys? Burnt sugar. You have to remember that word, burnt sugar. So burnt sugar like odor will be there in the urine. So burnt sugar like smell will be there in the urine, and that's the reason why it is said to be the maple syrup urine disease. Maple syrup urine disease. Is it okay? I hope this is done, guys. So by this we are done with the glycine, alanine, valine, leucine, isoleucine. Glycine, alanine, valine, leucine, isoleucine has been completed, sir. ठीक है जी? Are you done with this one here? So that completes the aliphatic amino acids. Saran, before slides has been deleted, Saran. That is gone already. Are you done? And someone was asking me a doubt. Hello. Someone was asking me about yeah, Divya. You were asking me, okay, sir, when the proteins are formed by amino acids only, why don't we take amino acids directly? Normally we do that. When we are eating food, in the food it will be directly proteins only. We can't do anything with that. But when you take some additional supplements, we'll be doing that. And I hope some of the students who are interested in doing exercise and all, they'll be taking additional supplement of protein, and in that you'll be having BCAs. BCAs are nothing but your branch chain amino acid, valine, leucine, isoleucine, and these BCAs are necessary for additional supplement while doing exercise or something like that. Is it okay? So yeah, of course we do take amino acids in the form of amino acids also, but not during our routine eating and all those things. Additional supplement. Okay, Divya? I hope your question is answered now. चलिए सर, अब हम बढ़ते हैं आगे. Let's go ahead. So I think now you're observing yesterday's topic was like a big topic, guys. Metabolism of aromatic amino acid, aliphatic amino acid, time pass हो गया sir. Glycine, alanine, valine, isoleucine, leucine हो गया. And now please write on the heading there as the metabolism of basic amino acids. I hope you remember, in metabolism of the basic amino acids, we are having like 3 basic amino acids: lysine, arginine, and histidine. Okay, so first of all, write down there as histidine. Okay, and remember the only one thing that you have to remember: histidine is the one which is going to form histamine. We come across this histamine, serotonin very commonly during inflammation. If you have learned pathology, आपने पढ़ा होगा. Okay, तो inflammation वगैरह वगैरह जहाँ पर भी आप पढ़ते हो, wherever you learn these things, asthma, hypersensitivity reactions, and all those things, you come across histamine and serotonin. So histamine is actually formed from histidine, and in your MC exam they have asked how is histidine converted to histamine? Remember, by decarboxylation, by removal of carbon in the form of decarboxylation. So histidine is going to form histamine, and in the presence by which process, guys? That is by decarboxylation. And once you are done with that one, welcome here right now, sir. The second one here right now is arginine. The second basic amino acid that you have to learn is arginine. Arginine function number 1, it comes in urea cycle, and I don't have to teach you, and you don't have to learn this. Urea cycle we studied yesterday, and we know that in the urea cycle, the last one, arginine, is the one which is going to break down to form ornithine back along with the release of urea. Ornithine and urea, है ना, in the presence of an enzyme known as arginase. तो arginine urea cycle में आता है. Number 2 function of arginine, it is also helping in the formation of nitric oxide. Nitric oxide. Nitric oxide. ठीक है. And this will function of the arginine is extremely important. Nowadays they are asking like hell in your MC exam, sir. Every time, every now and then they are asking, sir. So remember, arginine is the one which is going to form your nitric oxide. Can anyone tell me what is the formula of nitric oxide? The formula of nitric oxide will be NO. And remember, it is formed in the presence of which enzyme? The name of the enzyme, your favorite one, NOS. And what is NOS? NOS stands for nitric oxide synthase. NOS stands for nitric oxide synthase. So please make a note of that. ठीक है? Is it okay? So please note that nitric oxide is actually synthesized from which amino acid? Arginine, in the presence of the enzyme nitric oxide synthesis. That's all. This is what you need to learn about the histidine and arginine, sir, and that completes about your basic amino acid. Don't worry about the lysine. Lysine I'll tell you wherever it comes. अभी नहीं, ठीक है? Lysine अभी नहीं, sir. Lysine I'll tell you afterwards wherever it comes. Now once you are done with the basic amino acid, guys, now let us go towards one last and final concept, but a very big concept in protein metabolism. Now my dear friends, please write on the heading there as megaloblastic anemia. Do you know megaloblastic anemia? Yes. Do you know megaloblastic anemia? Megaloblastic anemia occurs due to deficiency of what? Yes. Listen here right now, sir. It's not only vitamin B12, even folic acid also. Yes. Look here. Megaloblastic anemia will occur due to vitamin B12 deficiency, and not only vitamin B12 deficiency, even the folic acid deficiencies. So either folic acid is deficient or vitamin B12 is deficient, it will lead to megaloblastic anemia. I want every one of you to listen to this concept. It's a bigger concept. Okay, it will take 10-15 minutes of time, and I want you all to listen this from the very beginning, from the starting. And please don't ask me again to tell this one. Why? Because you cannot repeat. Today someone was asking me formation of T3 T4, can you repeat, sir? See, I told you yesterday 110 times कि जो है ना, that's a big concept, important concept. स्टार्टिंग से सुनिए, प्लीज सुनिए, क्योंकि हम लोग ना वो बार बार रिपीट नहीं कर सकते. If you have some doubt, I can clarify, but repeat the entire concept, it's not possible. That's the reason I'm telling you before itself. Okay, before itself I'm telling you, megaloblastic anemia occurs due to vitamin B12 deficiency or folic acid deficiency. It will take 10-15 minutes of time. Listen this from the very beginning. Okay. सबसे पहले as a doctor, okay Rahul, agreed. सबसे पहले एज अ डॉक्टर आपको पता होना चाहिए विटामिन B12 डिफिशियंसी मेगलोब्लास्टिक एनीमिया होता है. फोलिक support the patient suffering with megaloblastic anemia, and I come to you. After coming to you, you have diagnosed me, and after diagnosis confirm, yes sir, you are suffering with megaloblastic anemia. After confirming, now starts the problem. Why? What is the problem now? After confirming that I'm having megaloblastic anemia, the next challenge in front of you is that identify right now this megaloblastic anemia in my body, it's due to vitamin B12 deficiency or folic acid deficiency. इसको हम कहते हैं डिफरेंशियल डायग्नोसिस. यू हैव डायग्नोस्ड डिसीज, आपने डायग्नोस कर लिया है मेगलोब्लास्टिक एनीमिया करके. अब आपको इन द पहचानना है. नाउ यू हैव टू आइडेंटिफाई वेदर इट इज ड्यू टू विटामिन B12 deficiency और folic acid deficiency. Whether it is vitamin B12 deficiency or folic acid deficiency. कैसे पहचानोगे? How will you identify? And to identify that, you need to learn a little bit of biochemistry. चलो, I'm going to teach you like 2 diagrams now, 2 concepts. Listen here very, very carefully. Concept number 1. Concept number one, we are going to deal with the metabolism of sulfur containing amino acids. Metabolism of sulfur containing amino acids. Yesterday I taught you one reaction, that is norepinephrine. Norepinephrine gets converted to epinephrine. Can anyone recall and tell me how? Norepinephrine is getting converted to epinephrine by, yes, methylation reaction. It's one of the best example, classical example of methylation reaction by addition of methyl group. And who is adding the methyl group? It is SAM, S-adenosyl methionine. After adding the methyl group, it is becoming S-adenosyl homocysteine. है ना? And now my dear friends, please stop writing and listen to the background story, the important thing right now. See, SAM is the methyl group donor. SAM is the methyl group donor. In many methylation reactions in our body. So SAM is actually the methyl group donor, and it is the one which is going to give what's a methyl group, CH3 group. So I repeat again, SAM is actually a methyl group donor, and it is the one which is going to give the methyl group in almost okay. So in almost every methylation reaction in our body, in biochemistry, whatever methylation reactions are there, in almost every methylation reaction, SAM is the methyl group donor. SAM क्या करेगा? Methyl group donate करेगा. ठीक है. Now guys, it's common sense right now. If SAM is the methyl group donor, if it is giving the methyl group every time, okay, it is necessary for us to even regenerate SAM. SAM is giving the methyl group every time, है ना? But we need to even regenerate the SAM back. We need to get the SAM back, है ना? उसको regenerate भी तो करना पड़ेगा. And to regenerate that, what you have to do is we have to convert this again to SAM back and apply common sense. How to regenerate that, sir? कैसे regenerate करें? Already SAM है back. उसमें वापस methyl group add कर दो. Understanding here? So keeping this concept in mind, let's regenerate SAM right now, sir. Come on, start. First of all, this S-adenosyl homocysteine will further break down to homocysteine. S-adenosyl homocysteine will break down to homocysteine. Don't have to worry about this reaction. Don't have to worry about the enzyme. And now welcome to the important reaction to be learned. Now this homocysteine is actually going to react with a substance, and the name of that substance is methyl tetrahydrofolate. I repeat again, methyl tetrahydrofolate. I repeat again, homocysteine reacts with methyl tetrahydrofolate. Methyl tetrahydrofolate. So therefore what is happening right now is that I am adding the methyl group back. So this methyl group is added back here. So therefore if you add the methyl group back, then what is going to be remaining? Tetrahydrofolate. Okay. And then after adding methyl group, homocysteine is going to become methionine. Wow. So when you see these kind of reactions in textbooks, it looks like very complicated. यार कुछ नहीं है. It's nothing. Very simple. SAM ने already methyl group दे दिया है. तो अगर हमको वापस SAM चाहिए तो क्या करना पड़ेगा? methyl group add करना पड़ेगा. And who is going to add the methyl group? Methyl tetrahydrofolate is going to add the methyl group. And after adding the methyl group to homocysteine, it becomes what? Methionine. हो गया. Now you people already know the method to make the name of the enzyme. चलिए सर एंजाइम का नाम बनाइए. We are actually transferring methyl group. So therefore, what is the name of the enzyme? Methyl methyl transferase, and along with that the name of the substrate, homocysteine. And guys, this requires a coenzyme, and the coenzyme will be your vitamin B12. Here comes the hero of the story. The homocysteine methyltransferase is helping in transfer of methyl group. And what is the coenzyme required? Vitamin B12 is required there. Okay. And now, no need to learn this. Methionine will now get converted to S-adenosyl methionine. Acha. Methionine is going to become what's a S-adenosyl methionine. Okay. And why did I give you the name as metabolism of sulfur containing amino acid? Recall yesterday's class. Sulfur containing amino acid will be your cysteine, methionine. So this cysteine, methionine are all sulfur containing amino acid. So simultaneously we are done with the metabolism of sulfur containing amino acid as well as the regeneration of SAMs. हो गया? Are you all done? Come on my dear friends, are you all done? Chalo sir, बढ़िया. Now once you are done with this one diagram here, I promised you I'm going to give you two diagrams. Basically we are actually now Titan here is asking, sir, where are you getting this methyl tetrahydrofolate? It comes from folate, and folate is folic acid. Folic acid is vitamin B9, and that is folic acid metabolism. ठीक है, चलिए अब हम बढ़ते हैं आगे. Now the last and final concept that I have to teach you right now here in this is propionic acid pathway. Propionic acid pathway. ठीक है, come on sir, write down. V for valine, leave one line in the middle. Then next is methionine, and then isoleucine, and then threonine. ठीक है, and in the middle of this somewhere here you write down there as odd chain fatty acids. I'll tell you what I'm trying to write down here, sir. See, this is one very famous thing in biochemistry. In biochemistry, this is known as VOMIT, vomit. Every biochemistry student and faculty will know this vomit, sir. V for valine, O for odd chain fatty acid, M for methionine, and I for isoleucine, and T for threonine. अगर आप ध्यान से देख रहे हो, whatever I wrote here in black color, it's that amino acids, and whatever I wrote here in red color, that's actually a fatty acid. So these are like amino acids and this is like a fatty acid here. ठीक है, but मैंने इसको साथ में क्यों लिखा? Why did I write it together? All this, sir, vomit, when they undergo metabolism, their end product is absolutely the same. When this vomit will undergo metabolism, their end product is same, and their end product is actually, yes, propionyl CoA. Their end product is propionyl CoA. Is it okay? Their end product is propionyl CoA. And my dear friends, here I'm going to teach are very much important. Propionyl CoA is now going to get converted to methyl malonyl CoA. Okay. In short, I read it like MMC. Methyl malonyl CoA. Propionyl CoA is going to form methyl malonyl CoA. How? With the help of an enzyme, and the name of the enzyme here will be, yes, carboxylase. Addition of carbon, therefore carboxylase. And you will already know what is the full name. The full name will be propionyl CoA carboxylase. What is the full name of the enzyme, sir? Propionyl CoA carboxylase. And my dear friends, this is important. Why? Because it requires coenzyme, and the name of the coenzyme will be biotin. It requires a coenzyme known as biotin. And I hope my students are already making a list. Till now I told you about the biotin coenzyme. Two enzymes I told you, sir. मैंने आपको ना दो enzyme बताए. One enzyme I told you in the very first class about pyruvate carboxylase. You remember gluconeogenesis? Pyruvate carboxylase. There I told you, and here. And you people have to make a list of this one. So this enzyme requires biotin. And finally, the last and final reaction here is methyl malonyl CoA is going to get converted to succinyl CoA. Succinyl CoA. And this is going to get converted to succinyl CoA in the presence of an enzyme known as mutase. And you people know very well what is the full name here. The full name here will be the methyl malonyl CoA mutase, MMC mutase. Methyl malonyl CoA mutase, MMC mutase. Methyl malonyl CoA mutase, methyl malonyl CoA mutase. And my dear friends, this is the second enzyme that will be requiring vitamin B12. That's all. And this entire pathway here will be the propionic acid pathway. Propionic acid pathway. Is it okay? Fine here. Now once you are done with this one here right now guys, अगर ये हो गया है तो अब आ जाइए सर, वेलकम here right now. Now I'll tell you the final conclusion here right now. Now I'll be telling you the final conclusion here right now sir. ठीक है ना? Fine. Now this entire कहानी I started from here sir. Okay, there are so many questions here right now guys. Okay, तो ध्यान से देखना. That entire कहानी actually started from here, and now I don't want you people to write down here sir. Please stop writing sir. Megaloblastic anemia, it occurs due to deficiency of vitamin B12 and folic acid. Megaloblastic anemia occurs due to deficiency of vitamin B12 or folic acid. Suppose there is a patient in front of you, and that patient is having megaloblastic anemia due to vitamin B12 deficiency. अगर उस पेशेंट में विटामिन B12 डेफिशिएंसी है तो फ्रॉम टुडे ऑनवर्ड्स माय डियर डॉक्टर्स, एनी टाइम इन योर एंटायर लाइफ, आई एम टेलिंग यू, नॉट ओनली फॉर एमसी एग्जाम, एंटायर लाइफ, आपकी ज़िंदगी में अगर आपने कहीं पर भी एक पेशेंट को देखा हैविंग विटामिन B12 डेफिशिएंसी, टुडेज क्लास शुड कम इन योर माइंड कि आजम सर ने कहा था कि देयर आर 2 एंजाइम्स रिक्वायरिंग विटामिन B12. विटामिन B12 कम्स हियर, होमोसिस्टीन मेथिल ट्रांसफरेज, एंड विटामिन B12 कम्स हियर, दैट इज MMC म्यूटेस. Vitamin B12 is required for how many enzymes? 2 enzymes. अब यहाँ पर देखिए सर, look at the screen. Please don't write. If the patient is having vitamin B12 deficiency, this enzyme is not working. I will not be able to repeat this sir. I'm telling you again and again. If there is a vitamin B12 deficiency, then this enzyme not working. If this enzyme is not working here right now, then what is going to happen? This reaction is not taking place. Then homocysteine levels will go up. Okay. So if the patient comes to me with vitamin B12 deficiency, उसके blood में, in his body, homocysteine levels will be increased. One thing. Number 2, if vitamin B12 is deficient, even this enzyme is not working, then this reaction is not going to take place. So therefore, methyl malonyl CoA levels will also go up. So therefore, yes, in his body, even MMC, methyl malonyl CoA, will also be elevated. Okay, fine here. Now megaloblastic anemia, patient number 2, another patient, but this patient is having folic acid deficiency. Now if this patient is having folic acid deficiency, my dear friends, I told you on the very first day itself, can I call the ic acids as 8 salts? Ic acids as 8 salt. Folate is the one which is going to form your methyl tetrahydrofolate. Folate is going to form methyl tetrahydrofolate. Hello my dear friends, what I am trying to tell you is, I am going to repeat here, folic acid, can I call it as folate? Hello, confirm me. Can I call the folic acid as folate? Yes, ic acids will form 8 salts, our basic principle. Therefore, folic acid or folate, it is the one which is forming methyl tetrahydrofolate. Oh my god. If there is no folate, folic acid deficiency, then there will be no methyl tetrahydrofolate. When there is no methyl tetrahydrofolate, there will be no reaction taking place. अगर ये reaction नहीं होगा, if this reaction doesn't take place, again homocysteine level will be elevated. Okay. So in this also what is going to happen is the homocysteine levels will be increased. Okay. Now please look at the second reaction here right now. In this second reaction here, my dear friends, do you find any role of folic acid here? यहाँ पर कहीं पर भी फोलिक एसिड का रोल है क्या? नहीं है. We don't have any role of the folic acid here. That means MMC level will not increase. Okay, in folic acid deficiency, MMC methylmalonyl CoA will remain normal. Therefore, welcome back. I am in the hospital. A patient comes to me with megaloblastic anemia. I am going to do a blood test. And in the blood test, if I find the homocysteine as well as methylmalonyl CoA elevated, then definitely I will tell as a doctor, yes sir, you are suffering with vitamin B12 deficiency. Beautiful. If a patient comes to me with megaloblastic anemia, I will do a blood test. In the blood test, there is only homocysteine elevated, not methylmalonyl CoA, only homocysteine elevated. Yes sir, you are suffering with folic acid deficiency. Are you understanding your point here? This is how you will be able to do the differential diagnosis in depth. Is it okay? Understanding here right now? So this, we have completed the concept of megaloblastic anemia, as well as we have completed the concept of sulfur containing amino acid, as well as we have completed the concept of propionic acid pathway. ठीक है ना? The end product of odd chain fatty acid, propionyl CoA, one of the question है ना? Biotin requiring enzyme, and we have done with the vitamin B12 requiring enzyme. So we have completed like many concepts and many questions in a very small manner, sir. Is it okay? Read me. Wow, चलो सही है, पूरा रामायण हो गया. And this person is asking who is Ramayan, who is Sita. Read me. The full form of MMC is methyl malonyl CoA. I have said n number of times during explanation. Methyl malonyl CoA, MMC. ठीक है सर? That's all about this concept here right now. That completes the entire protein metabolism also, sir. Protein metabolism topic completed, sir. Yesterday that we have started with the protein metabolism, ho gaya, done. So end of this, what you have to remember, guys, what are the important substances obtained? Everyone look here, I'll tell you. Just listen here right now, guys. Melanin, yesterday we have studied. Melanin is obtained from tyrosine. T3 and T4 is obtained from tyrosine, and then catecholamines are also obtained from tyrosine. And apart from that, melatonin is obtained from tryptophan, serotonin from tryptophan, as well as one more thing, that is vitamin B3 is obtained from tryptophan. Nitric oxide we studied today is obtained from arginine, and then we have studied about histamine. Histamine is obtained from histidine. Okay, so in this protein metabolism, you need to learn which amino acid is giving you which important substance. So which important product is obtained from which amino acid. Is it okay? Fine here. So you are going to remember that in the biochemistry part. Galaxy S10 पता नहीं क्या नाम है. What are the symptoms of megaloblastic anemia? Sir, you will be studying the symptoms and how to treat that and all those in your other subjects like pathology and all. Okay, so that is not my thing here to teach you about the symptoms and treatment of all that in the biochemistry part. वो आप बाद में पढ़ोगे. That's a big story. Yes, homocysteine increases in both the conditions, Ajay, and how to differentiate both the conditions is by only MMC. Chalo. So everything is clear here right now. So that completes this topic here right now, guys. I'll give you 2 minutes of time. I want you to first of all revise everything has been done till here, sir. तो एक बार इसको revise करो. Then we'll start with the lipids topic today, sir. We'll finish off the lipids topic today. Is it possible both folic acid and vitamin B12 are deficient at the same time? Yes, there is a possibility. ठीक है. Divya is asking limit dextrin is converted to linear chain. What is the name of the linear chain? Linear chain का नाम कुछ भी नहीं है मैडम जी. Linear chain का नाम linear chain ही है. It is just a linear chain, no name for that one. चलिए. Now I hope all the doubts has been clarified and let us continue with our discussion right now. ठीक है. Now the next thing. Now the next thing that we are going to learn. ठीक है. Sir, if there is a biotin deficiency, Bharat, थोड़ा control करो भाई. Whatever has been done, please ask the doubts in that one. Don't worry about that one. Biotin deficiency and everything we'll study in vitamins. We have let off vitamins topic. Sir, anatomy session करवा दो. Anatomy session is already done, right? चलो. Prajal is asking most abundant amino acid. Obviously, it is glycine. Glycine is most abundant. Chalo, sir. Now, once you are done with this, proteins wala topic right now. Now, let's start with the lipids, and we are going to learn the lipids. Okay, we are going to learn the lipids in exactly the same manner as we have learned yesterday. Okay, na? Arjun is asking RBC maturation is a symptom of megaloblastic anemia. Arjun, I told you symptoms, signs, treatment, everything afterwards in other subjects like pathology and all. Which are odd chain fatty acid? iPad, control, have some patience. I will tell you about fatty acid in lipids. ठीक है, चलो हो गया. Now let's get started with the lipids topic, guys. पहले मैं आपको बताऊंगा ना normal chemistry of lipids, and then I'm going to teach you about the metabolism of lipids. Okay, so please write on the heading as chemistry of lipids. Let's begin with the chemistry of lipids. ठीक है. iPad is saying I cannot see chat. Yes, iPad, I have disabled that. Only you can chat with me. Some people were doing the cross comments, so therefore I stopped that. Okay, so just chat with me, only me. Next one, chemistry of lipids right now, guys. Licosa lipid. First of all, let us start again the topic from the same thing, from the scratch. कि यार lipids होते क्या है? What are lipids actually? ठीक है ना? See here, lipids are actually na. Now you might be finding this definition something like more of chemistry part that is very much weird. Okay, chemistry of lipids. Okay, no problem. Now are you able to listen to me? I think there is a lag, it seems. Yes, everyone here? Now you're, yeah, thank you. Now chemistry of lipids. Lipids are actually the esters of fatty acid and glycerol. Esters of fatty acid and glycerol. Sir, what is the meaning of this so different? In organic chemistry, one of the basic principle in organic chemistry. Okay, one of the basic rule in organic chemistry, guys. I think you have learned about organic chemistry. In organic chemistry, one of the basic rule is that carboxylic acids, when this carboxylic acid reacts along with alcohol, these two will react together to form ester. And this reaction is known as esterification reaction. इसको हम कहते हैं S with the release of water, and there is esterification reaction. Carboxylic acid will be COOH, alcohol will be OH, and ester will be like C double bond O and O. ठीक है. What happened, guys? Is there a still lag? Are you able to listen to me now? Are you all able to listen to me now? Okay, now, okay, fine. Listen here, sir. See, this is one basic principle of chemistry. There's a basic principle of chemistry that carboxylic acid will be reacting with alcohol, and whenever these two react with each other, carboxylic acid and alcohol reacts with each other, it will lead to the formation of what's a ester. Now, carboxylic acid formula will be COOH, alcohol formula will be OH. When they react, ester form होता है. Fine. And now, my dear friends, out of all this carboxylic acid, we are going to take which carboxylic acid? We are going to take the fatty acid. Okay. Out of all the carboxylic acid, we are going to take which carboxylic acid? Fatty acid. Okay. So fatty acid is one of the carboxylic acid. Out of all the alcohols that you have, acetyl alcohol, methyl alcohol, ethyl alcohol, out of all that alcohols that you have, we are taking glycerol. Which alcohol are you taking, sir? Glycerol. And definitely when these two will react together, it will lead to the formation of ester. And remember, this is not like something normal ester, it's known as lipid, something special, lipids. I hope now you're able to understand. I repeat this again, guys. Are you able to listen to me now? Everyone here? Are you all able to listen to me now? Just confirm me. Yes. Now I'll repeat this again for you people here. Listen here right now, guys. In our school days, we have learned a basic principle in organic chemistry. Carboxylic acid reacts with alcohol. Whenever carboxylic acid reacts with alcohol, it will lead to the formation of ester. Carboxylic acid with alcohol, it will lead to the formation of ester. Out of hundreds and hundreds of carboxylic acid, fatty acid, out of hundreds and hundreds of alcohol, glycerol. So when you react them, obviously what is the basic rule? They have to form what? Only ester, and this ester which is formed here is lipid. So what is a lipid, guys? Lipid is the ester of fatty acid and glycerol. So from today onwards, do one thing, please make your life easy. Any time in any subject, if you listen the word as lipid, two words should come into your mind, sir. One is fatty acid, another one is glycerol. Two words should come into your mind, fatty acid and glycerol. Lipid बोलते ही आपके माइंड में दो वर्ड्स आने चाहिए, fatty acid, glycerol, fatty acid, glycerol. ठीक है? And now let me just add some more information here right now, guys. If suppose there is one glycerol molecule, just a schematic diagram. If suppose there is one glycerol molecule, on this one glycerol molecule, remember there will be three fatty acids attached. Lipid, yes, I'm having fatty acid and glycerol, lipid, and these lipids are also known as triglycerides. Lipids are also known as triglycerides. Hello everyone, now are you able to listen to me? Yes, Saran. Now are you able to listen to me right now, guys? There is one molecule of glycerol, and on this one molecule of glycerol, remember there are totally three molecules of fatty acids attached. On one molecule of glycerol, there will be three molecules of fatty acids attached. That's the reason why fatty है ना? So first learn the name, guys, the lipids or triglycerides or fats. क्या हो रहा है? What is happening? Why there is a suddenly lag in the middle? Everything was going good now. Okay, no problem. Okay, guys. Just confirm me if it is perfect now. Yes, everyone here right now, sir? So what I was trying to tell you is that okay, on one glycerol molecule, on one molecule of glycerol, there will be three fatty acids attached, and that's the reason why that lipid is also known as triglyceride. On one and now, my dear friends, our today's topic is all about fatty acid. There is nothing much to study about glycerol. Glycerol is just, it's an alcohol. Glycerol is just an alcohol. Nothing much to study about glycerol. And now this topic is all about the fatty acid, guys. Okay. If you remember, like yesterday's topic was about the proteins. But in proteins, remember we have studied everything regarding what? Amino acids. Similarly, today's topic is like lipid, and in lipid we need to study everything about the fatty acid, guys. So in the lipids, everything we need to study about what's sir? Fatty acids. So this topic is all about fatty acid. So please write on the heading there as